you're next. Okay, now I see you. Okay, very brief introduction. Katia Zolotovsky, I hope I don't mispronounce your name, is joining us from Boston, but she's actually at the Rhode Island School of Design. She's an architect, a biologist, and a biodesigner. She even designed Bacteria. She works at the intersection of science and design, which is always a very interesting area. Again, if you want the full bio, go to lasertalks.com. Okay, all yours.
Thank you, Piero. Can you see my screen the right way?
Yes.
Okay. Great. Let me just arrange it real quick. Okay, great. Hi, everyone. I'm happy to be here. I'm connecting from Boston. I'm here to introduce engineered living materials, which were the subject of my PhD and postdoc work at MIT. My work at MIT was focused on growth as a spatial and computational process. I was based in the design and computation group in the architecture department. I'm architect and biologist by training. And I worked on NSF-funded research in the Department of Material Science, and then for a postdoc in the Center for Synthetic Biology. That's where the design of bacteria comes in. I am now an assistant professor at the Rhode Island School of Design, where I continue to bridge science and material innovation with art and design. So engineered living materials, what are they? Biological systems assemble tissues and structures with great properties in ways that cannot be achieved by man-made materials. Living materials self-assemble under mild conditions, are autonomously patterned and self-repair and sense and respond to the environment. So inspired by this, the field of engineered living materials aims to use genetically engineered organisms to generate novel materials, meaning that in ELMs, we have materials that contain living cells with responsive function with some kind of polymeric matrix that has the scaffolding structural function, and thus can be designed as active and responsive biomaterials. So biological organisms are experts in harvesting energy from the environment and using the information stored in DNA to assemble molecules in the environment into materials and quite large structures like elephants and trees and humans. The technology of synthetic biology, which is a relatively new discipline, emerged at MIT in the beginning of this century, makes the computation of living cells accessible for non-expert biologists like us, creating designable, modular, predictable genetic components that introduce new behaviors into living cells. So we can have this power to grow materials, products, and maybe even architecture. Designers and architects become increasingly more engaged with the technology of synthetic biology. This is an image from a conference at MIT Media Lab on wearable biotech and global interfaces in 2020, right before the pandemic. Scientists, engineers, and designers came together to imagine the future for biotechnology beyond the field of medicine. They envisioned how we could integrate ELMs, or living bits of material, into materials, products, computers, devices, and even augment our own bodies. So those living bits of material with embedded computation that can sense the environment, process, and actuate the response. Living cells as simple as bacteria, yeast, and even synthetic cells can be programmed to sense light, heat, pathogens in the environment, and output tangible things like smells, color, potentially also translated into digital signals. My own work with ELMs is focused on this bacteria that carries the DNA instructions to assemble sugar molecules in the environment into cellulose fibers, a pure form of the same cellulose that is the main component of plants, trees, and many of the textiles we use. You can see here on this image that there's tiny nanofibers extruded through the membrane as bacteria swim in nutrient-rich liquid and rapidly grows. This is a little video of the process, how the fibers are assembled into bundles of fibers and then into a sheet. We are looking from the side. So the sheet of material is generated on the top as a top layer on the surface of the liquid. The biological material production happens in room temperature and can be inexpensive, as research has shown that bacteria can be grown on the sugar-rich waste from food industry, and nutrients can be provided by a symbiotic culture with other organisms. You can see how the cellulose material accumulates on the surface. This is the white stuff. It can be grown and harvested in large quantities to create garments and other products. So this material system is very appealing as the material can be produced in large quantities and the bacteria cells stay embedded in the cellulose matrix and can be programmed to sense and respond to the environment. You can see here from the left to right a microscopic image of the bacteria cells embedded in the cellulose matrix under microscope, the wet material being harvested, and then a larger scale sheet molded on a wooden panel and dried. Many designers has used this material as a leather-like material substitute for leather, like sustainable alternative for leather. Here you can see the bio jackets by Susan Lee, who pioneered design work with this material. Leather-like shoes created by molding and layering the cellulose sheets. This material can also be tanned, laser cut, dyed, very much like leather. Here are some examples of dyeing the material in situ or as it grows using natural dyes. Here you can see work by Moya Hawk, who proposed growing the material in the shape of a pattern to eliminate the fabric waste, which is a huge problem in the fashion industry. However, the designs I showed are only concept designs, since some properties of this material prevent it from being a viable option as it's grown. For example, the material is highly porous and water absorbent. It absorbs 95% of its weight water. So if it rains, the jackets will dissolve. So my process was concerned with hacking the material system at the cell level and finding ways to edit the material outcome, both in properties and the shape. So my goal through the process of scaling up was to explore the possibility of also keeping the material alive and responsive. On the nano, I proposed introducing new genetic programs into the bacteria. And then on the mesoscale, shaping, patterning, harvesting, and processing the material as it grows. And on the larger scale, I built a bioreactor as a design element that allows designers to interact with the growth process. So hacking the material production by introducing synthetic gene networks. How do we do that? So bacteria has, in addition to the double helix DNA, it also has those tiny circular plasmid DNAs that synthetic biologists and biotechnology in general use to write new genetic programs on. So some areas, so they built those synthetic gene networks, meaning that some parts of the DNA are responsible for production of molecules and proteins. Others are responsible for regulating this process of material production. So shutting it on and off or linking it to presence of some signal in the environment. This is a screenshot from a software that is used to design those small DNA molecules. You can see on the right the sequence of the 4 letters. And then you can send it out to an external company. This is how we did it in the lab. And you will get a tiny vial with a new DNA program. So this slide describes the protocol for introducing the newly written DNA program into the cellulose producing cells. The process is like all biotechnology method is manipulating drops of liquid and using heat or electricity to create holes in cell membranes to accept the new DNA programs. So what kind of programs do we want to introduce to the cellulose producing bacteria? This slide shows a research from a decade ago in synthetic biology of creating synthetic gene networks for photographs. So each cell on a Petri dish acts like a pixel and decides based on the rules whether to produce ink or not. So basically, the synthetic gene network introduces decision-making into cell linked to production of ink. My goal was to use, in the lab, was to use the same logic of computational logic linked to material, editing of material properties. So for example, making the material hydrophobic, so producing molecules that will turn the cellulose into hydrophobic, or increase the density of material locally. So patterning of material properties. I spent quite a long time in the lab. However, successes were some. There is another group, luckily, there is always, when it's interesting problem, there is colleagues working on it. So there is a group in Imperial College that is gradually making more and more successes in building genetic library for this material system. And you can see on the right here, this is a co-culture with yeast that does allow some kind of patterning of the biofilm of the cellulose. So this is work in progress. So my work also focused on the larger scale where I feel comfortable. So on the intermediate scale, we developed this process of shaping the cellulose using this natural growth property of cellulose to grow between liquid and air on the interface, and shaping this interface using silicone that is air permeable but not liquid permeable. So by creating those growth vessels, we can shape the cellulose in enclosed shape. This allows us to keep them moist with liquid inside and keep them alive and growing. So then, using this method of shaping the cellulose, we built a bioreactor system, meaning scaffold for growing the material, but also keeping it alive. You can see that each growth vessel has inlet and outlet, and we can regulate the flow of materials and liquids in and out of the growth cells. In this way, regulate the growth of material. So this is the whole system. You can see the growth vessels in the middle. We also have vessels for nutrients and property modifiers. And all of this is under digital control, where we can write the code and regulate the flow of liquids. This is extensive infrastructure that we needed to control this bioreactor design. Some more pictures of it. So this was proposed for a responsive facade for architecture that allows us to kind of connect to existing liquid flowing through the building, gray water and such. I have an ongoing project, a collaboration with UPenn. So what we do now is we're trying to eliminate the need for this extensive infrastructure and embed the responsiveness in the material itself. So we work also with a CellFree system developed by synthetic biologists to be more accessible for us to use, that have already programmed input and output. And you can see on the slide some of the outputs, fluorescence, colorimetric proteins, smell, change of material properties. All of these outputs are already programmed in the CellFree system. So I collaborate with Laia Mogas-Saldivila from UPenn and Vlasta Kubusova, who is right now a Fulbright Fellow in UPenn. And we are thinking how we can create, so what Lai is an expert on large-scale fabrication with water-based biomaterials. So we are developing a system that allows us to embed the responsiveness of genetic circuits into the material and then 3D print larger structures. So this is a slide that summarizes some of my collaborators. So you can see that my work spans architecture, synthetic biology, and material science, the Ortiz Laboratory at MIT. And my collaborators are architects, synthetic biologists, material scientists, and I also had a PhD supervisor in history of science, Sophia Rust. Yeah, and this research was funded by NSF fund that I initiated and applied for. Okay, I will stop sharing.
You are also an architect. What's the contribution of architecture to the work you presented?
Yeah, so as architects, I mean, my group that I worked in is about computational methods in architecture. So for me and many others, there's already growing a community of people in architecture working with living cells. This is just another way of introducing computation, right? So this is rule-based, and then we can, by simple rules, we can generate complex system, complex patterns. So it's basically following up on or generative computation that was previously very much popular in our field in architecture, but with actual, materials. So instead of simulating, so there is a lot of like algorithms we use in architecture that simulate biological growth. To me, now we are doing the real thing, right? So we can imagine how we can use cells to actually grow architectures, maybe small scale in the beginning, but who knows, maybe many of the small things can assemble into larger things like swarm robots.
Have you had any interest from the industry for practical applications that can, trickle down to ordinary families?
So actually, yes. So this material, the bacterial cellulose, I worked with a collaborator and we started together and we applied for an NSF. I mean, she's leading this effort. She's a synthetic biologist and she applied for NSF SBIR grant for starting a company. So now, and we received it. So now there is a company named Spatial that is developing genetically engineering this bacteria to grow material for, we started with architecture applications. This is how I spent my summer, like talking to many people in industry for a best application for this material. We switched to fashion just because the price, it's very hard to compete with architectural materials in terms, because they can produce a lot very cheap. Yeah, so right now it's the second phase for the NSF funded startup.
Mm-hmm. So this research is mainly done at the MIT, your research at the MIT, or where is physically your lab? Is at the MIT now?
So everything I showed today was done at MIT. I finished there 2 years ago, and now I am assistant professor in Rhode Island School of Design, where I explore how research can be done in an undergraduate liberal arts institute.
Are there other groups in the world that do something similar to the MIT project you described?
Definitely, yes. So there is a field of engineered living materials that I refer to, and it's a growing field. There is several review papers that came out in Nature and Science, kind of outlining this field. However, most of work is done on the synthetic biology scale of genetically engineered engineering cells is mostly done for medical applications. I think for the reason that's where the funding is coming from. And traditionally, and this is a very important field, of course, but taking those methods and techniques and thinking how we can apply them to products in architecture, I think is a very important new field.
Excellent. Okay, thank you. Thank you very much.